Ever feel like your life is just one big, non-stop game of Tetris?
Uh-huh.
Trying to get everything in the right place at the right time.
Right.
Well, get ready to be amazed because today we're diving into the world of cellular logistics,
Okay.
where the stakes are even higher and the organization is infinitely more impressive.
Yeah.
We're talking about how cells manage the movement of thousands of proteins,
Right.
those tiny workhorses that keep everything running.
It's true.
You've shared some fascinating lecture notes on this.
Yeah.
And trust me, this is way cooler than watching a perfectly synchronized Rube Goldberg machine.
What's mind-blowing is that for a cell messing up, even one delivery could be disastrous.
Oh, wow.
Every protein has a specific job, a specific place it needs to be.
Right.
It's like an elaborate ballet, but instead of dancers, we've got enzymes receptors. Uh-huh.
And signaling molecules all moving in sync.
So how do they do it?
Yeah.
It's not like our cells have tiny little postal workers directing traffic.
Right.
These notes mention something called signal sequences. Is that the secret sauce?
Absolutely.
Okay.
Think of signal sequences like those address labels you slap on a package.
Right.
These short stretches of amino acids are attached to proteins
Okay.
and tell the cell's transport system exactly where each protein needs to go.
Okay, so it's like a zip code for proteins.
Yep.
What happens if a protein signal sequence is wrong or missing? Is it like sending a package to the wrong address?
Yeah,
Chaos in the mailroom.
you got it.
Okay.
And there's a really cool experiment with the VSVG viral protein that demonstrates this perfectly.
Okay.
Scientists tagged this protein with a fluorescent marker.
Basically, they could watch it move around the cell,
Sure.
but when they messed up its signal sequence,
Yeah.
the protein got stuck in the ER,
Oh, no.
like a package lost in transit.
Remind me what the ER is again.
The ER, or endoplasmic reticulum, is like the cell's manufacturing and assembly plant.
Right.
It's a network of membranes where proteins are produced,
Okay.
folded into their proper 3D shapes, and often get their first modifications.
Got it. The protein factory.
Yeah.
And the VSVG experiment showed that without the right address label, proteins can't even make it out of the factory.
Precisely.
Yeah.
And scientists actually use similar techniques like radioactive labeling
Uh-huh.
to map out these cellular delivery routes.
Oh, wow.
It's like attaching a tiny tracking device to a protein and following its every move.
That's incredible.
Yeah.
Speaking of tracking the notes, also mentioned fluorescent protein tagging.
Right.
Is that like a live feed of these cellular deliveries?
Exactly.
Wow.
While radioactive labeling gave scientists a good overall map,
Uh-huh.
fluorescent tags were like switching to GPS tracking in real time.
Oh, cool.
They could watch proteins moving from the ER to the Golgi apparatus,
Okay.
which acts like the cell's post office,
Okay.
and beyond.
So we've got a protein factory, the ER,
All right.
and our packaging and shipping center, the Golgi.
Yeah.
But not all proteins are needed at the same time, right?
Right.
Some are in constant demand, while others are more like emergency supplies.
Yeah.
How does the cell handle that?
That's where the cell really shows off its logistical genius.
Okay.
With two different systems.
Okay.
Unregulated and regulated secretion.
Right.
Unregulated secretion is like the steady flow of mail delivery.
Right.
Proteins are constantly being produced and shipped out.
Yeah. Okay, makes sense.
Yeah.
What about regulated secretion?
Right.
That's the emergency stockpile system.
Exactly.
Okay.
These proteins are held back,
Right.
stored in special containers until the cell receives a specific signal.
Yeah.
It's like having a fire extinguisher.
Right.
You don't want it going off randomly,
Right.
but you definitely need it ready to go at a moment's notice.
And what kind of signals is the cell waiting for?
It could be anything from a change in nutrient levels to a message from another cell.
Oh, wow.
Think about insulin, for example.
Okay.
Your pancreatic cells constantly produce insulin,
Right.
but it's only released into your bloodstream when your blood sugar levels rise after a meal. That's regulated secretion in action.
Wow. It's like the cell has its own internal intelligence system,
It's true.
constantly monitoring and responding to changes.
It is.
It makes you appreciate how much is going on behind the scenes to keep our bodies running smoothly.
Absolutely.
Wow.
And we're just scratching the surface here.
Oh.
And those address labels we were talking about, those
Yeah.
signal sequences?
Uh-huh.
Well, proteins can actually get additional shipping instructions along the way.
Really? Tell me more about that.
One crucial process is glycosylation.
Okay.
This is where sugar molecules, yes, actual sugar, are
Uh-huh.
attached to proteins,
Right.
often while they're being processed in the Golgi apparatus.
I thought glycosylation happened earlier in the ER.
That's what scientists initially thought.
Yes.
But these notes highlight a cool discovery.
Okay.
It actually starts in the cis Golgi,
Okay.
a specific compartment within the Golgi apparatus.
So the Golgi is more than just a shipping center.
Right.
It's like a multi-level processing plant.
Precisely.
Wow.
Think of the Golgi like a factory with different departments.
Okay.
The cis Golgi is the receiving dock.
Okay.
Then you have the medial Golgi, where proteins get modified and sorted.
Uh-huh.
And finally, the trans Golgi acts as the shipping department.
And glycosylation, those sugar additions, what's their purpose?
Right.
Are they just decorative, or do they have a function?
Those sugars are far from just decorations.
Ah.
They're like adding special instructions to those address labels.
Right.
Glycosylation can affect a protein's folding,
Uh-huh.
stability, and even how it interacts with other molecules.
So it's not just about getting the protein to the right place,
Yeah,
but making sure it's in perfect working order when it gets there.
exactly.
Yeah.
And sometimes those sugar molecules act like a signal to send the protein to a very specific destination.
Okay.
The lysosome.
Oh, right, the lysosome.
Yeah.
We touched on that earlier.
Yeah.
Like the cell's recycling center, right?
It's more than just recycling.
Oh.
It's the cell's ultimate waste disposal and recycling plant combined.
Oh, wow.
It's filled with enzymes that can break down virtually any molecule the cell no longer needs.
Wow.
And one way a protein gets tagged for delivery to the lysosome is through a specific sugar
Okay.
called mannose-6-phosphate.
It's amazing how the cell uses something as simple as adding a sugar molecule to direct a protein's fate.
Right.
But we've been focusing so much on what the cell makes
Mm-hmm.
and how it transports it.
Yeah.
What about things coming into the cell?
Right.
That's a whole other logistical challenge. It's like
Yeah.
we've been so focused on the outgoing mail,
Yeah.
all the packages and things going out.
Right.
But what about the incoming packages and deliveries? How does the cell handle that side of things?
That's where endocytosis comes in.
Okay.
The cell's way of bringing in everything from nutrients to signaling molecules,
Right.
even fighting off invaders.
Okay, so endocytosis is how stuff gets into the cell.
Right.
But we're talking about a completely different set of processes
Yeah.
here, right?
Exactly.
Okay.
And just like with protein production and sorting, the cell has multiple ways of doing this,
Ah.
each tailored for different types of cargo. Imagine a cell needs to take a sip of its surroundings,
Right.
maybe to sample what molecules are floating around.
Right.
That's penocytosis,
Oh.
bringing in small molecules and fluids.
So penocytosis is like the cell taking a quick taste test.
Yeah, you could say that.
Oh, okay.
Yeah.
What if it wants something more substantial?
That's where phagocytosis comes in.
Okay.
Imagine the cell needs to engulf a whole bacterium or a large chunk of debris.
Right.
Phagocytosis is the heavy lifter, using its cellular membrane to surround and engulf larger targets.
So penocytosis is for sipping and phagocytosis is for gobbling.
Yeah.
But no matter how it gets in, it all ends up in the lysosome eventually.
Yeah, you got it.
Okay.
But it's not a direct route.
Okay.
Before anything gets broken down in the lysosome, it usually makes a stop at the endosome.
Yeah.
Another sorting station within the cell.
Wait a minute. Another sorting station?
Yeah.
Is the cell that afraid of mixing things up?
Well, you have to admire the cell's organizational skills.
Right.
Remember how we talked about the Golgi being like a multi-level processing plant?
Yeah.
The endosome is similar.
Go.
It receives incoming vesicles from endocytosis and sorts their contents, deciding what gets recycled back to the cell surface and what gets sent to the lysosome for demolition.
Okay, so it's like a triage center for incoming deliveries.
Yeah, like a triage center.
What determines whether something gets recycled or sent to the lysosome for disposal?
A lot of it has to do with molecular tags and signals.
Okay.
Remember those sugar molecules we talked about?
Uh-huh.
The ones involved in glycosylation? They can also act like flags, marking certain molecules for delivery to the lysosome.
So those sugar tags are like putting a sticky note on something that says, lysosome, please dispose of properly.
You could say that.
Right.
Yeah, the lysosome is like the cell's recycling and waste disposal center all in one,
Right.
breaking down molecules into their building blocks, which the cell can then reuse.
Talk about efficient.
It's very efficient.
But what about the cell's own components?
Right.
Imagine things wear out eventually.
Yeah.
How does the cell handle its own internal spring cleaning?
That's where things get really interesting.
Okay.
The cell has this remarkable process called autophagy, literally meaning self-eating.
Whoa, self-eating, that sounds a bit intense.
It sounds more dramatic than it is.
Oh!
Autophagy is a highly controlled process where the cell identifies its own damaged organelles or protein clumps,
Right.
things that are no longer working efficiently,
Right.
and breaks them down for recycling.
So it's like the cell has its own internal renovation crew,
Yeah.
identifying what needs to be demolished and carted away.
Precisely.
Wow.
During autophagy, the cell forms a special compartment
Okay.
called an autophagosome, which engulfs the targeted components.
Yeah.
And then guess where that autophagosome goes?
Let me guess, to the lysosome.
You got it.
That seems to be where everything ends up eventually.
That's right. The autophagosome fuses with the lysosome, and those powerful enzymes go to work breaking down the old components into reusable building blocks.
Right.
It's like the ultimate form of cellular recycling.
Wow. It's incredible how much is going on inside our cells that we're completely unaware of.
Right.
From protein production and trafficking to waste disposal and recycling, it's like a bustling metropolis operating with incredible precision.
That's true.
And on that note, I think this is a good place to wrap up this deep dive.
Yeah.
A huge thank you for joining me on this journey into the fascinating world of cellular logistics.
Of course.
I hope you're leaving with a sense of awe and wonder at the incredible machinery that keeps us alive and ticking.
It is pretty amazing.
Until next time, keep exploring.